Good afternoon. I'm Kevin Grady, the Senior Product Line Business Manager at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series. Today's webinar, entitled iPSC-derived primary cells, expand your cell-based assays with an unlimited and biologically relevant resource, is presented by Dr. Brian Shapiro. Brian Shapiro is a scientific content specialist for cell biology at ATCC. In today's webinar, Dr. Shapiro discusses how differentiated induced pluripotent stem cell-derived cells fulfill the need for a consistent source of cells and physiological relevance. He will then provide data demonstrating how differentiated iPSC-derived cells can be incorporated into immunoassays or further differentiated into other cell types, such as osteocytes, chondrocytes, and adipocytes. If you have any questions for our speaker, please use the chat function available through the webinar program. Questions will be answered as time allows at the end of the presentation, and any remaining questions, as well as the recorded webinar presentation, will be archived on the ATCC website at www.atcc.org. So with that, I would like to welcome Dr. Shapiro.
Thank you, Kevin. Before I jump into my talk, I'd like to say a quick word about ATCC. Founded in 1925, ATCC is a nonprofit organization with headquarters in Manassas, Virginia, and an R&D and services center in Gaithersburg, Maryland. We are the world's largest, most diverse biological materials and information resource for cells and microbes. We are also an innovative R&D company featuring advanced in vitro models such as iPSC-derived primary cells, which is what I will be discussing today. So today I'll start by talking about how to select the appropriate cell line for your experiments. I'll quickly run through where differentiated induced pluripotent stem cells, or iPSCs, lie in the continuum of cell models. Then I will give some background about iPSCs. I'll go into ATCC's portfolio of iPSC-derived cells, showing you their differentiation potential and some application data. I'll then end my talk with a wrap-up of the presentation and take your questions. So let's jump in. First, I'm going to discuss the pros and cons of the different cell models and where ATCC's new iPSC-derived cells fall. ATCC has a large variety of classical and advanced cell models. We developed these advanced cell models to enhance our offerings and continue to be a complete solution provider. Most people are familiar with ATCC for our continuous cell lines such as HeLa cells and A549 cells. But as you continue down your research path, you will need more biologically relevant cell types. And ATCC provides these in the form of primary cells, H-tert immortalized primary cells, and now iPSC-derived primary cells. All these cell types feature ATCC's cell line reliability, authentication, and performance. So like I mentioned, I first wanted to talk about selecting the appropriate cell model for your research. As a researcher, one of the first questions you have to ask yourself is, which model is the best for my workflow? This table breaks it down for you. Let's start with continuous cell lines. They have high proliferation capacity, and they are sturdy cells that are easy to work with, but they are tumor-originated cells, which make them less biologically relevant. They might have chromosomal aberrations or mutations or things like that basically make them lose some of their physiological relevance. By contrast, primary cells are isolated directly from tissue, so they have the greatest biological relevance. However, the supply for primary cells is relatively low from a given donor, and there can be significant lot-to-lot variability due to biological variance between the donors. Now next, there's H-tert immortalized primary cells. These cells are genetically modified primary cells. H-TERT cells exhibit normal primary cell physiology, but have the growth characteristics of a continuous cell line. They have near normal genetic stability. They have the ability to generate a large number of cells for large drug screens or toxicology studies, providing limited to no donor variability. And then last, the star of today's presentation, iPSC-derived primary cells. They significantly mimic in vivo tissue type physiology and genetic stability, and we can generate a great number of production lots from an individual clonal source, which highly limits their lot-to-lot variance. Now, just to mention, as a complete solutions provider, ATCC offers mesenchymal stem cells in primary or H-TERT immortalized forms. These are isolated from adipose tissue, and also an iPSC-derived version from bone marrow. We offer CD34 positive cells in primary and iPSC-derived, isolated from bone marrow, and we have monocytes in primary form, isolated from peripheral blood, and iPSCs derived from foreskin fibroblasts. The differences here are the primary cells will need to be used immediately and are not amenable to passaging. The H-TERT immortalized cells can be passaged to over 25 PDLs, or population doublings, post-thaw. They're derived from one donor and thus show reduced interexperimental variability, like I mentioned before, and they represent one solution. However, the H-TERT immortalized solution isn't available for each cell type. iPSCs are another solution. As I mentioned, they're also derived from a single donor, but either can be terminally differentiated or used right out of the vial. And, as I mentioned, these will exhibit the greatest interexperimental reproducibility. So I've talked about where iPSCs fall in the research workflow. Now I'll introduce iPSCs and talk about what iPSC-derived cells are and what they can do for your research. iPSCs, which, as I mentioned before, stand for induced pluripotent stem cells, are derived from somatic cells that have been reprogrammed back to an embryonic-like pluripotent state. These iPSCs can be subcultured indefinitely as long as differentiating cells are removed from the culture. Alternatively, iPSCs can be terminally differentiated to any cell type via various culturing methods with lineage-specific media formulations. Any cell type is possible, from ectodermal lineage cells, such as neurons and keratinocytes, to mesodermal lineage cells, such as hematopoietic and endothelial cells, to endodermal lineage cells, such as thyroid cells and the acinar, ductal, and endocrine pancreatic cells. To generate iPSCs, ATCC introduces the reprogramming factors OCT4, SOX2, KLF4, and MYC genes into somatic cells using a Sendai virus integration method. This method is footprint-free, producing a final product that does not contain any viral DNA. The method provides a renewable source of pluripotent stem cells that can then be turned into the cell type of your choice. While iPSCs are useful in and of themselves, many researchers prefer to have their cells in a ready-to-use format. We've also heard many toxicologists and cancer researchers were challenged by the lack of a consistent source of cells with high physiological relevance for their cell-based assays. The source tissue for primary cells comes from a variety of donors, which can introduce interexperimental variability and confound experimental results. Cell lines, by virtue of the fact that many of them are from neoplastic tissue, may not have the physiological relevance needed to make the assay predictive. So ATCC heeded this call and produced cells that answer all of these needs. So let's talk about the star of today's show, iPSC-derived primary cells. Like I mentioned, ATCC wanted to create a versatile cell type that could be produced in great quantities, yet retain physiological function. We looked to in-house produced iPSCs as feedstock for this project, as they were, one, available, and two, we have over a decade of experience culturing these cells. For differentiation, the iPSC-derived cells were differentiated to the desired cell type via incubation in proprietary media formulations. Also, since we used in-house produced iPSCs, we can supply the parental iPSCs, which can be used as a control if needed. The major benefit of using a single clone source to generate each of these iPSC-derived primary cells is that you have the potential for an unlimited, consistent source of cells, which you can use for large-scale research applications, whether it be research or therapeutic purposes, and suffer no donor variability. For the first phase of the portfolio, ATCC decided to focus on 3 cell types: mesenchymal stem cells, CD34 positive progenitor cells, and monocytes. To bring us all up to speed, I thought I'd go through some definitions. So let's start with mesenchymal stem cells, or MSCs. These are multipotent stromal cells that can differentiate into a variety of cell types, including osteoblasts, or bone cells, chondrocytes, or cartilage cells, myocytes, or muscle cells, and adipocytes, which are the fat cells that give rise to marrow adipose tissue. Next, we have CD34 positive cells. These are hematopoietic stem cells that can differentiate into the cellular components of blood. For example, erythrocytes, or red blood cells, leukocytes, or white blood cells, and platelets. Finally, we have monocytes. Monocytes are a type of leukocyte that can differentiate further into macrophages and dendritic cells. Now let's take a deeper dive into ATCC's new iPSC-derived differentiated primary cell portfolio. The first product I'd like to talk about is iPSC-derived mesenchymal stem cells, ATCC part number ACS-7010. These cells are derived from ATCC's bone marrow iPSCs. They show high purity, as 95% or greater of the cells are positive for a number of markers such as CD29, CD44, CD73, CD90, CD105, and CD166. These are checked by flow cytometry. Less than 5% of the cells are negative for CD14, CD19, CD31, CD34, and CD45. In addition, less than 5% of the cells are positive for the iPSC marker TRA160. Now, these cells do exhibit high post-thaw viability at greater than 90%. Since they come from a single clone, they will be available in large quantities. And they also show high osteocyte, adipocyte, and chondrocyte differentiation potential. I should note that we are using serum-free media to cryopreserve these cells. So as I mentioned, we use flow cytometry to monitor the surface markers of the iPSC-derived MSCs for QC testing. These data show that expression. On top of the standard MSC markers of CD73, CD90, and CD105, we test for CD29, shown on the upper right, CD44 on the upper middle, and CD166 on the lower middle to meet International Society for Cellular Therapy guidelines. In order to further characterize that over 95% of the initial starting material differentiate into MSCs and not any other cell type, our release testing includes MSC negative surface markers. And here I'm showing you that. Very low expression of CD14, CD19, CD34, CD45, and TRA160. By the way, this information is also provided in the lot-specific C of A. As I mentioned before, MSCs have the potential to differentiate into mesodermal lineage cells such as bone, cartilage, and fat cells. The next couple of slides show this. Here, our iPSC-derived MSCs were differentiated to osteocytes using ATCC's osteocyte differentiated tool, part number PCS-500-052. We did this for 37 days and stained using alizarin red, which stains bone cells. Undifferentiated MSCs are shown at the left image on day 37, and differentiated stained cells are shown on the right. The red staining indicates the presence of MSCs differentiated into osteocytes. Here, iPSC-derived MSCs were differentiated to chondrocytes in 3D culture using chondrocyte differentiation tool, which is ATCC part number PCS-500-051. For 21 days, we did that. The cells were fixed, sectioned, and stained with Alcyon blue stain, which dyes cartilage. I used the control from the previous slide for comparison so you can see the profound blue stain indicating the cartilage in the extracellular matrix. In this slide, iPSC-derived MSCs were differentiated to adipocytes using an adipogenesis differentiation kit. We did this for 21 days and stained using oil O red stain, which stains fat cells. Undifferentiated MSCs are shown in the image on the left, and differentiated and stained cells are shown in the image on the right. So these data indicate that the iPSC-derived MSCs can be differentiated into 3 different cell types. MSCs have also been reported to immunosuppress activated peripheral blood mononuclear cells. ATCC has previously shown that with bone marrow-derived MSCs. Here, either iPSC-derived MSCs or primary bone marrow-derived MSCs were co-cultured with peripheral blood mononuclear cells, or PBMCs, at different ratios and evaluated for CD45 PBMC using flow cytometry. Red bars represent iPSC-derived MSCs, and the green bars represent bone marrow-derived MSCs. As you can see in the control, where there are no MSCs, most of the lymphocytes are proliferating and are expressing CD45. When the PBMC to MSC ratio is 1:1, most of that proliferation is arrested, and this is seen in both the bone marrow and iPSC-derived MSCs. Even after increasing the PBMC to MSC ratio, you can still see that PBMC proliferation is inhibited. I'd like to point out that lot-to-lot variability was significantly higher in the bone marrow-derived MSCs than the iPSC MSCs, indicated by these error bars. This supports our claim that iPSC-derived MSCs provide more consistent results or better interexperimental consistency. So I've showed you that the iPSC-derived MSCs can differentiate into fat, bone, and cartilage cells, and they can also immunosuppress activated PBMCs. So now I'll discuss the second product in the portfolio. The second product is iPSC-derived CD34 positive progenitor cells, which are derived from ATCC's bone marrow iPSCs. The part number is ACS-7020. These cells show high purity and post-thaw viability. They are suspension cells and are available in large quantities. As CD34 positive cells, they show high erythroid, myeloid, and megakaryocyte differentiation potential. They're also cryopreserved in serum-free media. So here's the flow analysis of the iPSC-derived CD34 positive cells. Positive markers that we test for are CD34 and CD45. For CD45, even though it's a positive marker, it needs to be below 80% to show that the cells are not differentiating too far. The negative marker Tra160 is a stem cell surface marker. You can see that CD34 is over 90% and Tra160 is less than 1% here, indicating the high purity of the cells. In the next study, I'm about to show you the differentiation potential of iPSC-derived CD34 positive cells. In this slide, I'm showing you the general path of differentiation that a hematopoietic stem cell can take once it's been treated with the right cocktail of hormones and chemicals. When incubated with the appropriate media, iPSC-derived CD34 positive cells differentiate into colony-forming unit that generates myeloid progenitor cells, aka CFU-GEMM. The GEMM, by the way, stands for granulocyte, erythroid, monocyte, and megakaryocyte. These further differentiate into a mixed bag of progenitors. Megakaryocytes, which give rise to platelet progenitor cells and platelets. BFU-E, or burst-forming unit erythroid, which lose their nucleus, produce hemoglobin, and differentiate into CFU-E, or colony-forming unit erythroid, which give rise to mature erythrocytes or red blood cells. CFU-GEMM can also specialize into mast cells. The cells can also be coaxed into becoming myeloblasts, which differentiate into the various granulocytes or other components of the immune system. And so I'm showing you some of that differentiation potential in this slide. Here, the CD34 positive cells were differentiated to erythroid, myeloid, and megakaryocyte lineages using a commercial kit for 16 days. Basically, this kit contains erythropoietin, insulin, and the other factors needed to send the cells down the appropriate lineage. The micrograph on the top left corner is the CD34 positive colony seeded at 15K. Top middle is CFU-GEMM, which generates the myeloid cells. Top right is CFU-GM, which are precursors to monoblasts and myoblasts and are derived from the CFU-GEMM. Bottom left is CFU-GEMM with hemoglobinized erythrocytes, and you can see the red of the hemoglobin. Bottom middle is CFU erythroid, derived from the CFU-GEMM. And finally, bottom right is the burst-forming unit erythroid. So, like the iPSC-derived MSCs, the iPSC-derived CD34 positive cells have high differentiation capacity. All right, I've showed you the first two products in the iPSC-derived portfolio. Here's the final product that we offer. The iPSC-derived monocytes. ATCC product number ACS-7030. These cells are derived from ATCC's iPSC foreskin fibroblasts. Like the other iPSC-derived lines, they have high purity and post-thaw viability. We see greater than 90% of the cells to be CD14 positive. Like the other iPSC-derived cells, production lots derived from a single clonal source provide for limited lot-to-lot variability. As you can see in the upper right-hand panel, these cells' growth mode is in suspension. In vivo, monocytes can differentiate into dendritic cells and macrophages, shown in the lower panel. Macrophages need almost no introduction. Macrophages are the large white blood cells produced through differentiation of monocytes. They're capable of detecting, engulfing, and destroying pathogens in apoptotic cells. They also can respond to inflammatory stimuli. Dendritic cells, on the other hand, are a type of bone marrow-derived leukocyte. They're the most potent type of antigen-presenting cell of the mammalian immune system. Their main function is to capture antigens and present the processed antigens to the T cells. They act as messengers between the innate and adaptive immune system. They can also be used for anti-cancer vaccine development. As the lower panel alludes, ATCC-derived monocytes show high macrophage and dendritic cell differentiation, along with high cytokine activation and a high phagocytic potential for macrophages. I'll be showing you that over the next couple of slides. So we differentiated the iPSC-derived monocytes into macrophages for 6 days using in-house macrophage differentiation medium. We also used maturation mediums. Here on the leftmost image, you can see the morphology of the macrophages on day 6. They have attached to the plates and flattened out. In the middle panel, the same macrophages were incubated with a Giemsa stain. Giemsa stain stains macrophages bluish purple, as demonstrated here. To test if these were functional macrophages, we wanted to see if they could eat something. We used a kit that can specifically detect phagocytosis and endocytosis with pH-sensitive fluorogenic dye linked to E. coli-derived bioparticles. The dye discriminates endocytosed from adherent and extracellular particles by turning red in the acidic environment of the phagosome. So we incubated the macrophages with the kit for 2 hours. On the right-hand panel, we see that the macrophages are glowing red, indicating that they phagocytize the particles. Oh, and also in the lower panels, we see marker expression that lends support to the presence of macrophages. We see macrophage markers CD163 and CD68 by flow, with a decrease in CD14 expressing cells. So the iPSC-derived monocytes can be differentiated into phenotypic and functional macrophages. ATCC's iPSC-derived monocytes also have high dendritic cell differentiation potential. To support this, we incubated the monocytes with a commercial dendritic cell differentiation kit and cultured them on attachment culture dishes. On the left, you can see the monocytes prior to differentiation. They have smooth surfaces and are largely in suspension. In the middle and right images are the morphology of dendritic cells on day 7 of treatment. They're showing the characteristics of immature dendritic cells, including a rough surface with multiple pseudopodia, and they've also become loosely adherent. And looking at the results of the flow, like before, we see a decrease in CD14 positive cells along with a concomitant increase in dendritic cell markers CD80 and CD83, indicating that we have indeed dendritic cells. So while it's nice that data shows differentiation, but I'd like to show you an example of how you could actually apply monocytes to something useful. So in recent years, there's been a movement away from using animal-based tests. A good example of this is the various tests for detecting endotoxins, aka pyrogens, in parenteral preparations and biologics. So these compounds can cause a robust immune reaction and fever, followed by severe shock and death. Now, historically, there have been 3 tests that have been used to determine the safety of these parenterals. The rabbit test is the first one I'll talk about. It involves subjecting rabbits to the biologic or parenteral and then monitoring it for fever or death. Bad for the rabbit. Then there's the limulus amoebocyte lysate or LAL method, LAL method, which requires the blood of the horseshoe crab. Either a gel clot is then detected, or more commonly now a change in color or turbidity is measured. Unlike the rabbit pyrogen test, the LAL assay is limited by being unable to detect non-endotoxin pyrogens. And also, since thousands of horseshoe crabs are bled for this per year, and there's probably about a 25% mortality rate of the horseshoe crabs would probably prefer to be left out of this. So finally, there's the monocyte activation test, or MAT assay, which uses the cryopreserved blood of several human donors. While it's much more humane than the other 2 tests, it could suffer from lot-to-lot differences in quality. So you have influencing factors such as lifestyle and genetic background that can differ significantly between the donors, and really we just can't control for that. So we hypothesized at ATCC that since human monocytes from mixed blood are used in the MAT assay, our human iPSC-derived monocytes could be used for conducting the test. And I'll show you that right here. So in the panel on the left, we show a mix of PBMCs. They're ATCC actually, part number PCS 800-011. And on the right, we show ATCC iPSC-derived monocytes. Both are treated with increasing concentrations of LPS. So LPS is a lipopolysaccharide. It's a bacterial cell wall component, and it's highly immunostimulatory. We measured the secretion of interleukin-6, which is a pro-inflammatory cytokine that monocytes secrete when activated by something like LPS. As you can see, the interleukin-6 secretion increases as units of endotoxin increase. Thus, the iPSC-derived monocytes can be used as a humane, inexpensive alternative for conducting the MAT assay. And as I showed you, they have high potential to be differentiated into macrophages and dendritic cells. All right, so let me go ahead and wrap up my presentation now. I've showed you that ATCC developed proprietary methods for the differentiation of CD34 positive cells, monocytes, and MSCs from normal human iPSCs. Something that I didn't mention before, the different ATCC iPSC lines have varied efficiencies for differentiation into distinct lineages. So we commercialized the iPSCs that differentiated the most efficiently. Differentiated iPSC-derived primary cells can provide an unlimited source of cells for your cell-based assays, as well as interexperimental reproducibility and high biological relevance. They exhibit primary cell functionality and genetic stability. I also showed you that iPSC-derived CD34 positive cells can differentiate to all blood lineage cells. I presented data indicating that iPSC-derived monocytes can differentiate to functionally active macrophages and dendritic cells. I also showed that activated cells can be utilized for developing a MAT assay. I showed you how iPSC-derived mesenchymal stem cells can differentiate to osteocytes, adipocytes, and chondrocytes, and they also display immunosuppressive properties. And finally, if you're interested in more information or in ordering these versatile cells, please navigate to www.atcc.org/differentiated iPSCs. Thank you. I'll take your questions now.
Thank you, Dr. Shapiro, for that excellent presentation. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. This session will be documented along with the recorded webinar presentation on the ATCC website at www.atcc.org.
Okay, it looks like we lost Kevin's audio right quick. You might just have to reconnect to it. So I'm just going to jump on the first question here. Can you talk about the longevity of iPSC-derived CD34 positive cells? And then what about the iPSC-derived monocytes from the same asker? So I can say that it's definitely not recommended to passage these cells. Don't try expand or freeze them. They were designed actually to be used right out of the vial, hence the high number of viable cells. So we ship them out with approximately 3 × 10⁶ cells per vial, which is 3 or more times what we normally ship. And that's for the iPSC-derived CD34 positive cells. For the iPSC-derived monocytes, they come 2 × 10⁵, I'm sorry, 2.5 × 10⁶, cells per vial. So to sum all that up, be sure to use these cells in experiments immediately after thawing. All right, and I'm still not hearing you, Kevin. Kevin, you may have to unplug and plug back in your headset, and then click on where it says use new hardware. So I'll just go ahead and jump on the next question. To follow up on this, do you have any recommendations for seeding density and thawing media? Well, this is really, as far as seeding density, that's going to be based purely on the customer's application. So without knowing that, I don't have a good recommendation. But we do recommend using RPMI 1640 medium with 10% FBS just for thawing the cells. All right, here, let me go ahead and jump on the next question then. Now, how about some questions about culturing the iPSC-derived mesenchymal stem cells? Okay, what is the seeding density for the ACS-710 mesenchymal stem cells? Good question. We do recommend seeding these MSCs 10,000 to 20,000 cells per centimeter squared post-thaw and post-culture. In addition, optimal seeding densities are critical for MSC differentiation depending on the application that you're using. Now, if you're using ATCCs, osteocyte or chondrocyte, differentiation kits, we do have pretty clear instructions as far as what seeding densities you want to use. So basically, use the seeding density that's indicated on the kit. All right, and I'm going to keep going until I hear Kevin pipe in. What is the optimal cell confluence when passaging MSCs? So 85 to 90% confluence is optimum. Do not go beyond 95%, or you'll see a rapid drop off in the cell's differentiation performance. Okay. What is the effect of passage number post-thaw on lineage differentiation? Okay, so like all primary cells, early passage mesenchymal stem cells, for example, P1 post-thaw or whatever, are going to have a much better differentiation potential than later passage mesenchymal stem cells. So we're talking passage 4 post-thaw. So it's really best to use them earlier rather than later. Okay, we've got another mesenchymal stem cell question in here. So this is someone who might be interested in ATCC's other mesenchymal stem cells, maybe. What other MSCs are available from ATCC? So great question. There is a table in the presentation that I showed adipose-derived mesenchymal stem cells and adipose-derived H-TERT immortalized mesenchymal stem cells. I actually missed a product in there. We also have human bone marrow-derived mesenchymal stem cells, and that ATCC part number is PCS 500-012. Now, these were the cells that were used in the experiment on slide 19. That was where we showed equivalent immunosuppressive activity with iPSC mesenchymal stem cells and bone marrow mesenchymal stem cells. Okay, we have another one here on the longevity of ACS-710 iPSC mesenchymal stem cells. Oh, okay. So what they want to know is how many population doubling levels and passages can these cells go? So iPSC-derived MSCs can be proliferated up to 20 PDLs, or roughly equivalent to going 10 passages. And here's another question about passaging. What dissociation reagent is recommended for passaging the MSCs? Is any special medium used? All right, so for passaging the iPSC-derived mesenchymal stem cells, you'll want to use trypsin EDTA that's qualified for primary cells. We have that. It's part number PCS 999003. We also have a trypsin neutralizing solution that pairs well with that. It's part number PCS 999004. So those can all be found on the ATCC website under Media and Sera. Okay, here's a big question actually that's just come in. Can iPSC-derived cells be used in COVID-19 research, and what are the applications? All right, so that's a really big question, isn't it? Recently, there's been about 15 papers suggesting or reporting the use of mesenchymal stem cells or some kind of stem cell therapy to lessen the effects or improve outcomes for SARS-CoV-2 infection. Right off the bat, I've got to make the disclaimer that our cells are for research use only. However, that being said, these could be used as standards in these experiments, since we can reliably provide a consistent product and lots of it, actually. Now, another thing that these cells could be used for studying COVID-19 or SARS-CoV-2 infection. Earlier, I showed the mesenchymal stem cell immunosuppressive capacity. So they could be used to study mechanisms for immunosuppression, maybe to evaluate the anti-inflammatory response to viral infection to the cytokine storm. Interestingly, this just made me think of this and kind of relates a little bit back to my graduate work. The angiotensin converting enzyme, or ACE2 receptor, has been identified on hematopoietic progenitor cells. And this is interesting. It's one of the molecular mechanisms that SARS-CoV-2 uses to get into the cell. So while we don't have data showing the expression on our iPSC-derived cells, it would be really interesting to see and add another application to the list of things you could use the cells for. Now for the CD34 positive cells, they could be also used as controls to investigate treatments for the cytokine storm. There's also some work being done using CD34 positive cells to help repair lung damage from the infection. So again, these cells could be used as controls for that purpose. Now, for the iPSC-derived monocytes, they could be used in any application requiring PBMCs. For starters, many inflammatory processes are regulated by dendritic cells and macrophages. So you could differentiate the iPSC-derived monocytes into these cell types and then use them as controls to look at immunosuppression, but from the other side of things, right? You can use them to look at the interplay between monocytes or macrophages and T cells and how it relates to treatments for SARS-CoV infection. Also, any of the products I talked about today could be used to test the safety of vaccines and other therapies. And again, you know, you can get almost any amount of these cells since they're of clonal origin. On the side note, ATCC, that is, has a variety of primary PBMCs, peripheral blood monocytes, mononuclear cells, and primary T cells. And these cells can be used to study the efficacy of novel vaccine candidates to activate those cells. And you can find all of these at www.atcc.org/cancer-immunology. Now, here's a few questions that all came in from the same asker. Are monocytes mostly suspension cells compared to macrophages? Besides the markers, is it possible to differentiate between monocyte stage and macrophage stage morphologically and based on adherence? And can we consider activated monocytes to be macrophages when they become adherent and polymorph? So yes, monocytes are typically in a suspension, whereas macrophages tend to attach to something when they're differentiated. You can look for phagocytic activity like I showed in the presentation. Some of the other substrates you could use to look at phagocytic activity could be either latex beads or apoptotic bodies, and you could get those created by incubating cells with styrosporine. You could also look at cytokine responses, chemotaxis, or invasion capability as well. There's a lot of commercially available kits that you can use to look at any of those things. Okay, now here is another question. Are all cell lines aneuploid? And that's kind of interesting. So not all cultured cells are. Generally, normal primary cells won't be. That would be cells that are either derived directly from, from an organism, and are at basically passage 0 or at very low passage, right? Many cell lines are, though. They tend to exhibit chromosomal aberrations the longer that they're in culture or they have been passaged. They're going to undergo genetic drift the more times that they're passaged. Now, regarding the iPSC-derived cells, these cells are not aneuploid. They have a normal karyotype of 46, either XX or XY, depending on the donor that we use. Okay, here's another question. Which adipose, bone marrow, or umbilical cord mesenchymal stem cell product differentiates most efficiently into chondrocyte lineages? Okay, well, that's a good question, but before this talk today, I looked over ATCC's 2019 ISSCR poster. That's the International Society of Stem Cell Research that we presented in 2019. So it looked like we tested the MSC differentiation capacity of 4 different iPSCs. The part numbers were ACS-1019, which is foreskin fibroblast derived, ACS-1026, 1031, and 1030, which were all bone marrow derived, each from different ethnic groups. And what we saw was ACS-1026 could differentiate significantly better than the others. So that's what we selected to commercialize and then for further differentiate. So basically, we didn't follow up on the differentiation with the MSCs that had the low, with the iPSCs that had the low MSC differentiating capacity. I mean, it just really didn't make sense for us to. So the iPSC-derived MSCs came from iPSCs that originally were developed from bone marrow CD34 positive cells. And all of the iPSCs were selected for commercialization based on their overall performance. All right, and the questions keep rolling in. The differentiation of iPSCs to CD34 positive cells is not completely homogeneous, since at least 20% does not express the CD45 molecule. Why is this? I like this question. This is interesting. The emergence of CD45 expression does correlate with a commitment to lympho-myeloid cell lineage. But what you see is once you have a higher proportion of cells expressing the marker, the cells overall just have gone too far along the path to differentiate to lymphoid type cells. So you kind of lose the myeloid differentiation capacity to turn into, say, erythrocytes or megakaryocytes. Okay, there's a question about transfection here. If we generate our own protein constructs that have our own generated mutations, how do we transfect iPSCs or differentiated cells with our constructs? So that's a good question. Presumably these cells are transfectable. I can tell you that the parental cell lines are. Probably about 2015, ATCC transfected the parental cells with GFP using our transfection reagent Transfex. So we brought in Transfex, we wanted to see what cell lines it worked in, and we transfected GFP into the parental lines and they glowed green. So if you're interested in learning more about this, we actually have a transfection web page. If you go to www.atcc.org/transfection. Okay. Can iPSCs be differentiated into motor neurons? My answer to this is yes and no. So the differentiated iPSCs that I showed you today, I don't think you'd be able to do that. But if you mean by just iPSCs, TRA160, NANOG, and SOX2 expressing iPSCs, our research and development group actually did that. We differentiated those cells to create neural progenitor cells, which you can buy actually on our website. We've got a complete system actually of normal and Parkinson's disease trilineage capable neural progenitor cells. By trilineage, I mean they can turn into neurons, astrocytes, and oligodendrocytes. So some of these are actually lineage marker labeled. And we also have an expansion differentiation media. We've got a ton of application data actually around this, which I presented in a webinar, in 2018 actually. Time flies. If you're interested in viewing this, let us know and we'll send you the link to the video so you can view it on demand. And the cells are available if you navigate to www.atcc.org/neuro. All right, and it looks like we've got time for one more question before we wrap up. Can we request iPSCs to be derived from diseased patient donors with certain mutations? Okay, so that sounds like a custom service. And yes, you can contact our custom services group via either your account number or the ATCC website at www.atcc.org/custom solutions for more information. And we're happy to talk to you about generating these models. At this time, we'll conclude our Q&A session. Thank you all for attending the webinar. Any questions that we didn't get to today will be answered this afternoon and posted with the video at www.atcc.org. For more information, or again, to order what you saw today, please go to www.atcc.org/differentiated iPSCs. So, coming attractions, we're hosting more webinars this month. On June 11th, ATCC microbiologist Brittany Tang will be delivering her comprehensive talk on understanding COVID-19, a global pandemic. And on June 25th, bioinformatician Tasha Santiago Rodriguez will present her talk entitled Viral Metagenomics and the Use of Standards from Biology to Clinical Applications. Thank you again for your time, everyone, and have a great day.